So you've learned medical immunology, and Miss Lei taught you that topics. And medical microbiology somehow overlapped with immunology, because at the beginning of the two courses, the two areas overlapped. The development of microbiology came together with medical immunology. And this topic will explore a microbial world and its interactions with humans. So we are learning microbiology, we are learning the microorganisms, but we are also learning how they cause disease in human and how our human body responds to microorganisms. So basically that's the major part of our course. Before we touch any material of medical microbiology, I would like to introduce a little bit about myself. So now I'm in China, as you used to be here in Zhengzhou city. Okay, let me change. Okay, so I'm here, and I hope that you guys can come back to China as soon as possible when the pandemic is gone. And can you tell me are you from? Where's your hometown? Someone in India? Here? Some of you might be in Pakistan, Bangladesh, Middle East, Sri Lanka. Great, I think it's here. Yeah, when I was graduate student, I have one of my good friends was from Sri Lanka, Iran. Okay. China. Someone in China. Okay. Alright, so it's a wide distribution of you guys. A lot of countries. And so basically, there is one fact that you might know. No matter where you are now, we are all from Africa. So here, Africa, especially the East Africa, are the hometown of everyone. So we call it the sweet home. We'll talk about this topic a little bit later, that we human beings all came from Africa. Okay. Little bit about myself. So my name is Huaixin Zheng, or in Chinese, Zheng Huaixin. And gender male. And my age, I won't tell you. A little bit about my education background. I got my doctorate degree in molecular and cellular biology in New York. The university name is Stony Brook University. And I got my bachelor degrees in China. So in college, I learned bioscience and also management. A little bit more. I'm kind of open-minded, as you can see through this list. I'm atheist and also kind of a scientist, because except for teaching, I also do research on microbiology. Liberal, straight, but I'm a LGBTQ supporter. And also I'm a feminist. So in general, I'm kind of open-minded. So that's a little bit about me. when in the future, when you are a doctor, probably you will need to figure out a lot of information about your patient. So that's the form shown on this slide. So that's the patient questionnaire. And you need to ask your patient a lot of questions to know better about your patient and know their medical history and everything. So basically that's my questionnaire, the answer to my questionnaire for you guys to know. Yeah, as I mentioned just now that for this first class, we'll just cover the first topic, introduction of medical microbiology. Have you guys ever learned microbiology in high school or in middle school in your country before you came to China? Maybe a little bit on biology, right? Somehow in somewhat on biology, right? Great. That will help us to learn microbiology because, yeah, the general contents are the same, but our medical microbiology will expand the contents a little bit more and will focus on part of biology. So that's microbiology. So before our real story, I'd like to give you a very brief introduction of the big history. So that's a history not a history of human, not the history of life. So we'll introduce the big history. So that's a history of everything from 13.7 billion years ago. I know a lot of you have watched the TV show Big Bang Theory, right? And Big Bang Theory is, yeah, it's American TV show, but the song of that TV show covers this whole history. And it begins at that time point, and everything become reality at that time, and it generates matters, it generated matters, energy, and space and time. So everything generated at that time point. And this process we called physics. And after that, over 300,000 years, the universe cooled down and the energy and matters formed. And finally it formed atoms. And you know, with atoms, the chemical reaction can form and more complex molecules can form. And this process we call chemistry. And with chemistry, molecules making up organism, biology part can form. And after a long time of evolution, those chemical molecules came together and formed life. And we don't exactly know what time life formed in the universe, but on Earth, we know the time is about 3.5 billion years ago. So at that time, life formed on the Earth. So from that point, we will begin the area of biology. And after a long time of life evolution from 3.5 billion years ago to 2 million years ago, and compared to billion and million years is very short time. 2 billion years ago, humans appeared in Africa. As you can see here, I spelled the word humans with S, which means that at that time there were many species of humans in Africa during the whole evolution. So that is why we call Africa the hometown of humans. And 70,000 years ago, we, the Homo sapiens, left Africa and spread it all over the world. So that's the beginning of Homo sapien colonization from America, from Africa to Europe, to Asia, to everywhere, to even North and South America. So that's only 70,000 years ago. And about 30,000 years ago, we human beings, we Homo sapiens, became the only human species on Earth. So all the other humans disappeared and they died, and we became the only humans. So that made us very lonely. Only 12,000 years ago, so that is agricultural revolution, Homo sapiens learned to grow crops. We learned how to settle down. We don't have to hunt or fishing every day. We could live in villages, and cities can form, and governments can form, and civilizations can form. So that is the recent 12,000 years. What happened? People's life didn't change a lot in over 12,000 years, about that long time. And only in recent 500 years, we enter the area of science. So that's the big change. With the development of science and technology, with the accumulation of knowledge, we human beings changed even more. And the lifestyle changed a lot, and the technology changed a lot. And if you look back, even over 10 years or 20 years of your life, you can understand that lifestyle now is different from the time when you were a kid. So through this big history, we just want to learn the points that we human beings are nothing special but a species of life on Earth. So there are a lot of different species of organisms, of lives on Earth, and we human beings are just one of them, but nothing special. We share common things with all forms of life on the Earth. So we share the common genetic material, we share similar gene sequence with all the other lives. So all life on the Earth are connected to each other, not only within the human society, but within the whole world, including other species of life. And in our field, in what we are learning now, microbiology field, different microorganisms, they are also connected to each other, and they are also connected to us, human beings. Okay, so before we learn the subject of our course, I want you to keep the 2 points to understand biology. First, we should look backward. So that's what we just learned about the big history. We learned the whole evolution history of life, and we learned what happened before life came to reality. And we also look over the whole ecosystem. So you stand high and you look the whole ecosystem. And we should know that in the whole system, different types of lives are connected to each other, and we are all connected to the surrounding environment. And we are connected to the water, soils, and also the atmosphere. And any factor can influence the human society, such as global warming. All the problems are the humans' problems. And when people learn biology, we should know the famous statement that nothing in biology makes sense except in the light of evolution. So if we understand evolution, we'll understand biology better and deeper. So what we are learning? We are focusing on microorganisms. Like in the name, you can see that what we are learning are with two key properties. The first one is that micro means that small or tiny, which means that we are learning the tiny things. This stuff we are focusing on are very tiny. We cannot see them with our naked eyes. We need to use microscopes or even electron microscopes to see those small creatures. And the other property is that they are organisms. So generally, in this course, we are learning microorganisms. But now, this concept expanded. And in our course, we'll also learn some macro things, macromolecules, which are not organisms. It's also covered in our topic. For example, we'll learn prions, we'll learn virus. So technically, prions and virus, they are not organisms. They cannot live by themselves. They cannot replicate by themselves. So those agents, those molecules or particles, they are not organisms, but it's also covered in our course because they can cause infectious disease. So this scale bar, this ruler, shows us how small those microorganisms are. For example, here, these are the animal cells. The size of animal cells are about 10 or 20 micrometers. So micrometer is you divide millimeter to 1,000 parts, and that one part is 1 micrometer. And animal cells are about 10 or 20 micrometers. And what we are learning here, bacteria, is around the size of micrometer. So they are very small. We cannot see them with our eyes. And even smaller particles, we are learning viruses. They are even smaller than bacteria. We cannot see them with microscope. We need to use electron microscope to study viruses. And when we learn prions, those are just protein molecules. But the protein molecules are folded in the wrong way, which can cause disease in both animals and humans. And these molecules are even smaller. And we can study with electron microscope, and sometimes we need to use X-ray to study those protein molecules. So this is what we'll cover in our topic, and that's the tools. Microscope, electron microscopes are the tools we can use to study microorganisms. The microbial world are very different from the microbial world is very different from the world we can see. The microorganisms are distributed widely everywhere. We can see it's worldwide, everywhere. No matter where you can imagine, there are always some type of microorganisms. Like they can in earth, deep in the sea, high in mountains, high in the atmosphere. And also diversity. there are a lot of types of microorganisms, and the other property is that they interact with human beings. They interact with other species of microorganisms. And the interaction between microorganisms with human beings is what we'll focus on in our topic of medical microbiology. So why distribution? Microbial life are everywhere, and they are with the longest evolution history. The very first type of life on the earth must be some kind of microbial form, either some kind of virus or bacteria, we don't know. It's a long time ago. And they are everywhere, in the air, in soil, in water, and they can live on animals, on humans. And here, even in the harsh environment, like in the ocean black chimney, which is very high temperature, over 350 centigrade, there are some form of life, some form of microorganisms living there. In hot springs, the boiling water, some microorganisms can live there. In great salt lakes, with a high concentration of salt, microorganisms can survive in those water. And in sulfur springs, when the pH is very low, they can survive. This is the wide distribution. Diversity of microscopes. According to the diversity of microbes, according to their genetic material, like either DNA or RNA, or either how the genetic material organized, microorganisms can be divided into several categories. For example, the first one, eukaryotic microbes. In this word, eukaryotic, EU means real. So eukaryotic means that in those cells, there is real nucleus. There is membrane-surrounded nucleus. And we'll learn a lot of fungi in this group, such as yeast. We can use the yeast to make bread, to make beer. Okay, another category is prokaryotic. Prokaryotic microbes are those cells without a nucleus. They have DNA as their genetic material, but the DNA is not enclosed in membrane structure. And in this part, we'll learn bacteria. And also we'll learn non-cellular microbes. They don't have cellular structure. So these we'll learn viruses. So at this time, special time in the year, during the pandemic, and we should pay more attention on viruses. And also, if we have time, we'll learn a little bit on non-microbiotic molecules. So these are the prions. Prions are just protein molecules, but it's folded in a wrong way. And they can cause different disease in both humans and animals, such as they can cause the mad cow disease in cow and scurvy in sheep. So that's the prions. This is a kind of like a family tree about life on Earth. On the left are the prokaryotic form of life. In this part, we have two groups. The first one is bacteria. This bacteria we call the eubacteria. So that's what we are learning in this course. They are real bacteria. And EU means real, so they are real bacteria. And some of them are pathogens. They can cause disease in humans. The other part, this brown one, we call archaea. So archaea are a special form of bacteria. So we can call them archaea bacteria. Those are the ancient form of life, which some of them are frozen in the earth. Until now, we don't have any evidence that they can cause disease in human. So basically, this part is not covered in medical microbiology. On the right, the green part is the eukaryotic cells. So it includes algae, fungi, and also animals, plants. All these are with a cellular structure and with real nucleus. So these are the eukaryotic form of life. And we will learn eukaryotic microbes. And in this part, we'll learn different type of fungi. Some of them are pathogenic, which can cause disease in human, which can cause infection in human. Some of them are beneficial to human. We can use them as tools to make food. For example, we can use yeast to make bread and use yeast to make wine and beer. And in eukaryotic microbes, the major property is that they have true nucleus. So which means that their genetic material, like DNA, is enclosed within a nuclear membrane. So this is the nucleus. It's surrounded by nuclear membrane. And in this part, in the membrane vesicle, there are DNA molecules. So this is a major property. And besides the nucleus, in the cytoplasm, they also have other organelles. For example, they have mitochondria, Golgi bodies, and also endoplasmic reticulum. So that's short for ER. So they have different types of cytoplasmic organelles. So this is the major properties of eukaryotic microbes. Okay. Another part we'll focus on are the prokaryotic microbes. As we mentioned that prokaryotic means that they don't have nuclear membrane or they don't have nucleolus. So in this group, we'll learn bacteria. So this is the model structure of a bacterial cell. You can see that these are the blue part are the DNA molecules. It's condensed in part of the cytoplasm, but not surrounded in membrane. And this formed a structure we call the nucleoid. So it looks like a nucleus, but not a real nucleus. They retain basic cell structure and metabolic enzymes. In this structure, in this cell, they contain the basic cellular structure and they contain the basic essential metabolic enzymes. In the cell, there is no mitochondria, no Golgi bodies, and no ER. Bacteria don't have those cellular organelles. But in bacteria, there are ribosomes. Ribosomes are the machinery to make protein molecules. So it helps mRNA to translate into protein sequence, the peptide sequence. And in this part, like we mentioned, we'll focus on bacteria or bacterial pathogens. And these are some famous bacterial pathogens we'll learn in future. This one, the first one, is called Streptococcus. Streptococcus, in the future we'll learn coccus means that those cells are like balls, round balls, and structure means that they line up like a line, like a strip. So these are the streptococcus, and this is the staphylococcus. And again, coccus means that cells are round, and staphylo means that they group together like grapes. So it's a different form of grouping. And this one is Salmonella typhi. It's a gram-negative bacteria which can cause disease in human. And we'll also learn a little bit on non-cellular microbes. So that's virus. We'll learn the viral pathogens including HIV, HBV, and also polio, HPV virus, all those groups. And you can see the property of virus is that they don't have cellular structure. A virus particle is just a very simple particle. It contains the essential nucleic acid, either DNA or RNA, in the center of the particle, and then this particle is surrounded by protein molecules, and this protein coat we call the protein capsid. And this can form the basic structure of a virus particle. In some viruses, there is an envelope covering the core structure. This envelope is from the host cell. So when a viral particle leaves the host cell or the human cell, it takes part of the cellular membrane, and that forms this envelope covering the viral core structure. So this is a virus. And during this pandemic, we should know that this pandemic is caused by coronavirus. It's a form of RNA virus. And here shows us two types of viruses which can cause disease in human. On the left is a chickenpox virus. Chickenpox virus is not lethal usually. When we are kids, if we are not immunized, the virus can cause infection in the kids, and the symptoms including like the rashes on the skin all over the body. It's not specific to any part, it's all over the body to form this red rash, red points. And after this infection, the chickenpox can be latent. They can hide in our body for a long time, even lifelong. So the virus can be hidden in our nerve cells, and for a long time they are kind of quiet. But when we are getting old, when our immune system cannot work well, they can cause another infection. That infection we call zoster virus. And when these symptoms came, the virus will infect us from inside our body. It came from the nerve cells, and then it can infect the epithelial cells of our body. So it's very painful when zoster infection happens. On the right is poliovirus. Poliovirus can cause infection through digestive tract, but finally it can attack our nerve cells. And after that, the muscle of the patient cannot develop well. And this is the virus we want to eradicate from the world, from the earth. There is already one virus we humans successfully eradicated from the earth. So that's the smallpox virus. And WHO wants to get rid of this polio virus in 2020, but it seems that it's not possible to achieve this goal. I think now we have 4 countries all over the world having polio virus infection. I mean the infection from the environment. Only 4 countries. The good news is that we have vaccine against this virus infection. And when in the future, when you become a doctor, please try your best to persuade your patients, persuade your families and friends to take any possible and beneficial vaccine to prevent any infection in the kids. And one of them important vaccine is a polio vaccine. It's pretty safe. No worry about it. Okay, so interactions with human beings. So this is the third property of the microbial world. We learned that the microorganisms are distributed everywhere, and they are on our skin, they are living in our body, and even in our cells. There is one theory that mitochondria is just one form of microorganism which invade the eukaryotic cells, and they evolved inside the cell and become mitochondria right now. And so basically our body is not for ourselves, but for a whole ecosystem. We have an ecosystem on our body, and we carry a lot of microorganisms on our body. But that you don't have to worry about it, because most of the microorganisms, including either virus and bacteria, they are not harmful. They can live peacefully with us. They never cause disease. So those parts are the major part at the bottom of this pyramid. So they can be friends. Sometimes they can be friends. They benefit our health. They can provide molecules. They can provide substances our body can use. For example, the bacteria in our gut can produce some vitamins. They can help us to digest food and help us to get nutrients. So those are the good bacteria, good microorganisms. And in the middle of the pyramid, some of the microorganisms, they can be friends most of the time. They live peacefully with our body. They don't cause disease most of the time. But sometimes here, maybe when our body is weak, or when they change their location on our body, they can cause infection. Those bacteria are potentially pathogenic, or we call them conditional pathogen, or that opportunistic pathogen. They don't cause disease all the time, but sometimes they can. And our material in medical microbiology focused on this small group on the top of the pyramid a lot previously. So those are the real pathogens. So those bacteria or those microorganisms can cause disease, can always cause disease in humans. So that's what we learned a lot previously. And now we still need to focus on this part because they can cause disease. But with the development of modern microbiology, we are working on these 2 parts more and more. And now we know that some infection, the conditional pathogens, when it happens, when they cause infection in our body, sometimes it's a big deal. It's not easy to treat, and it can cause a lot of trouble. And also we need to learn the common flora and the friend microorganism in our body. They interact with us and they influence us. So that's one of the hot areas we are working on, or scientists are working on, microbiology. And we know that we are not alone. And in our body, we have microorganisms everywhere. In our respiration system, in our digestive system, on our skin, and even in our cells. The microorganisms are everywhere, and most of them are living peacefully with us. And similarly, in the environment, for example, for this small plant, there is a little shoot on the ground, but you can see that underground, the whole root system is very big, and microorganisms are living with the root system. It helps the plant to get nutrients from the soil, from the air. For example, some microorganisms can help the plant to get nitrogen, to fix nitrogen. There is one number probably you have no idea that on our human body, we usually have like 10 to the 13th cells, the human cells in our body. So that's the level of our own cells, 10 to the 13th. But on our body, the bacterial number is over 10 to the 14th, which means that we have 10 bacterial cells to 1 human cell in our body, which means that we are not living for us. We are bacterial carrier. We are carrying them everywhere. We are feeding them, and of course, in the other way, the bacteria also feeding us. They are helping us. So each human being is a pile of cells, is a whole system, whole ecosystem. Okay, we'll have like a 10 minutes break, and after 10 minutes, we'll come back to our lecture. Okay. If you guys have any questions or concerns about our lecture, you can just leave a message, and we can discuss a little bit during the break. And because for this first time, we won't cover a lot, but we'll finish this introduction part. So just now we learned that the microorganisms are living with us, are everywhere, and even on our body. We carry a lot of bacteria, we carry a lot of viruses. And we know that the microbes are interacting with the host. I mean, in human, that's us. They are interacting with us. And they are also influencing the evolution of host, like evolution of human and evolution of every species in the world. And most of them are friends. Microorganisms are friends to us. And some of them, very small part of them, can cause disease in humans. So we call them foes, enemies. Okay, with the left, sometime the rest time we'll talk a little bit about infectious disease, the impact of infectious disease on human. Right now, with the modern medical technology, with the development of modern hospitals, with the usage of antibiotics, we can handle a lot of infectious disease, especially bacterial infection. But in the history of human, before the era of science, before the modern society, the average lifespan was very short, mainly due to infectious diseases. For example, at the Bronze Age is about it's over 2,000 or 3,000 years ago, from 6 or 5,000 years to 2 or 3,000 years. So at that time, people learned how to make bronze, so we call them Bronze Age. The average lifespan was only 26 years, so that's very short. When people are young and strong, they cannot survive any longer because of some kind of infectious disease. And after that, even in the medieval Europe, so that's from 400 to 1500 AD, the average lifespan was only 30 years, still pretty short. About 100 years ago, so that's early 20th century, the lifespan was 50 to 64 years. So that's after the development of modern medicine. The lifespan becomes longer, doubles the amount of medieval Europe. And now in the whole world, the average number is 67 years. So a little bit longer than 100 years ago. And in developed countries, this number is much larger, much bigger. For example, in like developed countries like in Japan, this number is over 80 years. And in the USA, in America, the average lifespan is 78. And we should know that in the developed countries, USA is the worst. So 78, this number is the worst in developed countries. You know the average lifespan in your country? Number in China is, I think, 76, somewhat around that. Do you know the average lifespan in your country? You can reply. 65. In which country? This morning, some students from India told me that the average lifespan is 69 in India. So Bangladesh is 65. Yeah, so there is still space to improve, to get better. And that will depend on you guys. When you are doctors, when you become professionals, you can help the country to move better and to help your patients to live longer. Okay, so that's it. From the average lifespan, the impact of infectious disease. And in the history, infectious diseases influenced the human society a lot. For example, the deadly Black Death. When Black Death came, every time it killed about 30% of the whole population. So that's a big number because 1 out of 3 people was killed in any Black Death. Now that's a big portion of the whole population. And it changed the human history. According to the record, several pandemics of plague by bacteria Yersinia, and with the help of antibiotics, and if the patient is diagnosed correctly, we can use antibiotics to cure the disease. And we can handle the disease. We can confine the disease and block the spreading of the disease. So this is Black Death. Another form of disease is smallpox. Smallpox is caused by a virus, and it's also a deadly disease. And it's dangerous to kids. When the kids are like several years old, and when infected with a smallpox, about 20 to 30 of the kids cannot survive. Before people know the cause of smallpox, and people try to prevent smallpox infection and try to make vaccines at that time. They don't know what are vaccines, but they try to try different methods to prevent smallpox. And there are ancient records in both China and India. People are trying to use the patient's clothes or the patient's scar, powder of the patient's scar, to give other kids so that they can develop mild disease rather than killing them. And those kids are immunized to smallpox. But this vaccine is not widely used, so it didn't help to eradicate smallpox. And in Britain, a doctor called Edward Jenner, he developed a cowpox vaccine, and that is very safe and useful. And with that vaccine, we human beings finally eradicated smallpox in 1979. So ever since 1980s, there are no smallpox cases all over the world. So we don't have any smallpox infections anymore. Like I mentioned just now, that another virus we want to eradicate is the polio virus. And hopefully in recent years, maybe 5-10 years, with the usage of vaccines, we can eradicate polio virus, finally. Okay, another virus we want to learn is the influenza virus. And influenza virus are the virus which can cause flu, because the symptoms is kind of similar to common cold, even though flu is quite different and more severe than the common cold. And sometimes influenza virus are very dangerous, more dangerous than we imagine. Like what happened 100 years ago. From 1918 to 1919, so that's the year of the end of World War I, there is a flu pandemic all over the world, which is called Spanish flu. Spanish flu was originally from North America, and the soldiers carried the virus from USA to Europe, and it spread all over the world. And during that pandemic, and people estimated that more than 15 million people died due to a flu. It's not a cold, it's due to a flu. And now every year we have a seasonal flu. So now is from like October to next year, early next year, is called a seasonal flu, the flu season. To prevent the seasonal flu, we can have a flu shot, a flu vaccine every year. I think I think the flu vaccine should be available in your country. So if it's affordable and if you have if you can pay for it, I would suggest everyone to have a flu vaccine every year. Especially you can give your parents or your grandparents a flu vaccine every year to help them and to keep them healthy. So this is of influenza. And in the future, we'll learn influenza virus in detail. We'll learn how they cause disease and how their genomes are organized. Okay, another pandemic is AIDS, which is caused by virus HIV, the human immunodeficiency virus. And the disease is called acquired immune deficiency syndrome. Before 1980s, people didn't know anything about HIV, but in early 1980s, some patients were discovered in North America, and those patients finally were diagnosed with a virus infection causing AIDS. And now the virus is spreading all over the world. Until now, we don't have any vaccine against HIV, and we don't have any medicine to prevent HIV spreading. We don't have any medicine to cure HIV infection. So that's not good. But there is still some development, some progression people made over recent 40 years, 30, 40 years, that with the antiviral therapy, with the help of different medicine, a group of medicines, we can inhibit the viral replication. We can keep the patient healthy for a long time, and their average lifespan is somewhat the same to a healthy person. But still, we cannot cure it. The virus is always in the patient's body. We can only inhibit it, but we cannot get rid of it. So this is HIV. And another virus we know is HBV. HBV infection is one of the major problem in China, in Southeast Asia, and also in Africa. And due to a lot of reasons, it can be transmitted through body fluids like sexual contact, and also it can be transmitted through blood transfusion. So when the patients get blood from in the hospital, and if the blood is contaminated, the patients will get infected. And the good news is that against HBV, we do have vaccines, and those vaccines are working pretty well. After the kids are born, immediately after the kids are born, the doctors will give them a shot of HBV vaccine, and that can help them and prevent them from further infection. So this is a major threat to the human society. So after learning these examples of infectious disease, we'll learn a little bit on the history of medical microbiology. We'll learn how medical microbiology developed at the very beginning, and we'll learn how the scientists contributed to this field. Okay, medical microbiology, it become possible when people can see microorganisms. And in the history, we human beings used microorganisms for food production. For example, we can use yeast to make wines, to make bread, and a lot of food. We can use microorganism, but people use that according to their experience. They didn't know the reason behind. They didn't know why they can do this work. And now we know that the hero behind those industry is microorganisms. And only after the invention of a microscope, people can study microorganisms. That is invented microscopes are invented by a person named Leeuwenhoek. And he's a scientist and also a businessman. And this is the device invented by him. It's very simple, but it can enlarge microorganisms several hundred-fold so that with naked eyes you can see the cell. So this is the very beginning structure of a microscope. And with the development of technology, now we are having better and better microscopes, and with better resolution, and we can see the small cells, we can record the small cells. And the other person, the other hero we will talk about here is Edward Jenner. As I mentioned, that Jenner developed a vaccine to prevent smallpox. So that vaccine is generated from a cowpox. Cowpox can cause like mild infection in human, but it's not lethal, and people survive through that infection and get immunized against smallpox. And with this vaccine developed by Edward Jenner, we finally eradicated smallpox. And this is a human hero. Edward Jenner was a doctor in Britain. Another hero, a big guy, is Louis Pasteur. Louis Pasteur was a French scientist, and he contributed a lot in the area of microbiology. For example, he proved that fermentation and putrefaction are caused by the growth of microorganisms. And also he disproved spontaneous generation theory. Before Louis Pasteur, for a long time people believed the theory that lives can form in a proper condition, in a proper environment. For example, if you have a pile of hays and you leave them there for a long time, and finally the insects and worms can generate from a pile of hays. And if you have a can of crops and cover it, and after some time mice can generate from that environment. Now we know that it's not possible, but before that, people believed in this theory, spontaneous generation, for a long time. And with scientific methods, with well-designed experiments, Louis Pasteur finally disproved this theory. He also invented methods to kill bacteria in liquids, and this method we call pasteurization. It's a very useful way to kill bacteria in liquids, such as in milk, in juice, and it's still widely used in industry right now. And he also worked on immunology and vaccine. Louis Pasteur developed vaccine against rabies and also developed a vaccine against anthrax. Anthrax is caused by a bacterial pathogen. And he also established an institute named after his name, the Pasteur Institute. which is still a famous institute in the world. It's in Paris. Another big guy is Joseph Lister. Joseph Lister was a surgeon. He was a doctor. And he was the pioneer of antiseptic surgery. He used a chemical called carbolic acid. Carbolic acid is another name of this acid is phenol, to sterilize surgical instruments and clean the air in the surgery room. And it's very useful. And with this antiseptic surgery, the patient's infection gets lower and lower, and it's getting more safe than previous surgeries. And with his technology, he used to had a surgery for the Queen at that time, and the Queen recovered. This is Joseph Lister. Another big guy is Robert Koch. Robert Koch was a scientist in Germany. He was also a doctor. He was a doctor in a small town, and there he started working on microorganisms. During his time, industry of dyes developed in Europe, and people invented a lot of organic molecules with color, so that people can use those dyes to give color to cloth, to clothes, to fibers, and that. And Robert Koch tried to use those dyes to give color to microorganisms, so that he could study them under a microscope. And it's very successful, and this route is very successful. After staining the microorganisms, after staining the bacteria, he could clearly see those small cells. He isolated anthrax bacteria, and also he worked on tuberculosis. So that's the pathogen for TB, MTB. And he later of his life he worked on cholera. So that's the spiral bacteria causing cholera. Vibrio. Robert Koch invented a method called solid media culture. So he added some agar to liquid media, so that the media become solid, and so that he could grow bacteria on the surface of the media. And with this technique, he could isolate and purify pathogen from a mixture and get pure culture. And this technology is kind of a revolution at that time. And Koch's another contribution to this field is he gave these postulates, which is called Koch's postulates. And this is a general method or guideline to identify a bacterial pathogen. So the first step, or the first point, is that the organism or the pathogen must always be present in every case of the disease. If some disease is notified, and within all the patients of the same disease, the same organisms must always be present. And then this organism must be isolated from a host containing the disease. And then you can grow the pathogen in a pure culture. And use this pure culture, you can challenge a healthy susceptible animal, and you give the pure culture to the animal, and the animal should develop the same disease and develop the same symptoms. Then the same organism must be isolated from the inoculated animal and must be identified as the same original organism first isolated from the originally diseased host. So generally, this is the guideline or the procedure to identify some pathogen. And when Koch gave these postulates, he focused mainly on bacterial pathogen. Even though these postulates are not perfect, but it's very useful to identify the bacterial pathogen. And after the golden era of discovering bacterial pathogens in late 19th century or early 20th century, scientists discovered another group of pathogens. That's the virus. You know, the history of virus is very short, only like around 100 years, but it can cause a lot of trouble if it gets spilled over to the human society. As we can understand through the pandemic, one simple virus can cause a lot of infection all over the world, and a lot of people are killed by this coronavirus. Another contents in medical microbiology we'll learn a little bit is the discovery of antibiotics. Now we know that antibiotics are the substances or molecules which can kill bacteria. The first antibiotic discovered is named penicillin, and it's from some fungus forms. And this antibiotic can kill or inhibit the growth of bacteria, and it was discovered by Alexander Fleming in Britain. And after that, more and more antibiotics were discovered. And with the help of antibiotics, we can handle a lot of bacterial infections now. But it also caused problems because with the pressure of antibiotics, the microorganisms or bacteria evolved faster, and more bacteria become antibiotic resistant. So when they are resistant to antibiotics, we cannot use antibiotics to kill them, to inhibit them. We need to find new ways. We need to find new antibiotics to cure the bacterial infection. Okay, so basically that's what we'll cover today for this introduction. And thank you for your attendance, and